So we come to the last talk of this afternoon, integrating postmortem anatomy and in vivo imaging. And Rainer touched, of course, already important question, what are we doing when we do not know the precise physiology, when we do not have so good expectations about the processes that are going on. And Simon will comment on other strategies, how to interfere structural functional relationships. So thank you.
Thank you very much. Thank you for organizing that course and inviting me. So I use this a little bit of a wrap up, and going from the very high resolution that was just shown in the last talk, I'm really taking a little bit of a step back, because what we now have is an integration of data that comes from different subjects, some of them living, some of them dead, so we cannot go into the very same high precision that has just been shown. But I hope I can convince you that still we can get a lot of useful information from that. So what are we after? In the end, we want to understand why a particular part of the brain, a particular patch of the cortex, is doing a particular thing, a particular computation. And in order to understand that, we need to acknowledge that whatever is happening, locally specific happening in terms of function, derives from two major factors. One is extrinsic factors, so the kind of input and output, the interactions with other areas that a particular part of the cortex can entertain. But the second point also is then the intrinsic structure, which is basically the local infrastructure of that particular patch. And it remains to be argued that a particular spot on the cortex is fairly much determined in what will happen there based on these two aspects. What other regions I can talk to, gain input from and put output to, and what's the local computation infrastructure. And so a lot about understanding brain organization is really merging structure, function, and connectivity. Now, there's a very easy scenario to do that, and that's in non-human animals. There we can use, for example, electrode recordings to record the responses to different stimuli, and then in the same animal, at exactly the same location, we can inject tracers to find the connectivity at the cellular level, and again, in the very same individual, we can then apply histological techniques such as myelin staining or cytoarchitectonic staining to identify different cortical differentiation. Now, this really is the reason why a lot of what we know about cortical organization derives from non-human primate work, and I think it was illustrated quite nicely by a lot of references also in the previous talks. But the problem is all of that is very invasive, so we cannot do that in humans. And it seems very likely that once we go beyond sort of sensory cortices, this knowledge from non-human primates gets less and less well transferable. So in humans, we do have a lot of tools to look at brain function at different resolutional scale up to the columnar level, but mainly nowadays still on the meso and macroscopic level. We get more and more information about brain connectivity from a lot of different techniques, such as resting state and diffusion imaging, but it seems that we kind of, at least for a while, neglected the third component, and that's the intrinsic, the local structure. However, I would argue that, oops, I expected something else here. Now, why is that important? It's important because microstructural areas should represent functional modules, and are thus the most appropriate topographical reference for regionally specific activations. Now, what do I mean by regionally specific activations? Is basically any effect you see in neuroimaging, because the opposite of regionally specific would be global. So really anything that brain mapping is about is localizing regionally specific effects. And given what I said earlier, that it's a local microstructure and the connectivity, then we would really want a need to reference these regionally specific findings to microstructural areas. On the other hand, macroanatomy and stereotaxic locations are not sufficient for an anatomical interpretation of neuroimaging findings. Now, note that I said interpretations because they are perfectly fine to describe where something is happening, but you need to allocate them to specific units of processing, to specific modules that have a particular infrastructure that's different from the infrastructure of the neighboring patch of cortex in order to build a sort of common interpretation of structure and function. On the other hand, macroanatomical terms like the superior temporal gyrus can be okay in order to describe where something is happening, but we know there's a high heterogeneity of microstructural features, of, let's say, infrastructural units on that particular macroanatomical region, so that it's not really sufficient for interpretation. Now, the most common solution to that problem is to use the Taylor-Rock atlas. Now, the problem with that solution is twofold. One, from the purely technical perspective, it's a fairly coarse sampling. There's very sparse indication of areal borders. There's no information on interindividual variability, and it's not in MNI space, which is a space that most of your neuroimaging data is in. Now, these are all technical problems and can be argued that they may be overcome. However, the most important problem really is the last one, that the histological labels you get from the Taylor-Rock atlas are very coarse approximations. So Brodmann did an actual histological investigation. Now he summarized his findings in a schematic map, and then about 80 years later, Taylor-Rock and Tenu, they had their individual slices through that one hemisphere of that old woman, and they just put the slices next to the schematic drawing of Brodmann and roughly said, well, where could that be? So it's really an approximation from an approximation. And in that case, it may be fairly valid to argue that the histological labels you get from any Taylor-Rock demon are basically worse than anything you guess yourself by just looking at the Brodmann map. So that most likely is not the most appropriate solution for labeling brain activations. Now, as has already been introduced this morning, probabilistic cytoarchitectonic mapping represents a new approach to charting the cerebral cortex that overcomes many of these problems. I will briefly review the process. So we start out with 10 fixed human postmortem brains, and these are then scanned to document the size and the shape of the brain. And then we do the histological processing in whole brain sections that are stained for cell bodies to allow cytoarchitectonic analysis. The 3D histological volume is then reconstructed based on the MRI volume, block face images, and the images of the histological sections. And we can do cytoarchitectonic analysis. So that's both classical microscopy, where people look through a microscope and identify features based on laminar structure, on cell packing density, cell size, shape, and so on. But we also do a statistical analysis based on laminar cell density profiles. The results are then transferred to the reconstructed sections, and the post-mortem data is spatially normalized into the MNI reference space. And the crucial thing here now is that this procedure allows us to bring post-mortem data, information that you can only get once you've cut up and stained a slice of the brain, with a reference space that is the same that is used in vivo imaging for fMRI, PET, structural MRI, and so on. So in that standard reference space, we now can compute probabilistic maps for each histologically defined area. And this bridges the gap between the post-mortem and the in vivo world. And this is not just another representation, another feature of brain organization in vivo imaging, but rather what's crucial is that we now do also have the bridge into anything else that can be done on the microscopic level. Think about, for example, as you've also heard earlier this morning, the multi-receptor mapping any sort of neurochemical measurements. So anything that is done on postmortem microscopic data, you can stain neighboring sections for cell bodies, and you can identify the same areas. And since we also do have a representation of these areas in standard MNI space, we can really use that to bridge different lines of research that otherwise would have remained fairly independent from each other. On top of that, we also have the bridge then to the non-human world. So where again, a lot of the work is also doing microscopy and cytoarchitecture, myeloarchitecture. So again, many of the findings in non-human primates and rodents are related to particular cortical areas, not to a macroanatomical location, not to a stereotaxic coordinates, but to a particular histological area. And again, now we do have a representation of histological areas now also in the same space that is used for in vivo imaging. Now, when we create these probabilistic maps, we get one map for each area that has been examined. This is the most precise representation of the information. So for each voxel of the brain, we now know what is the likelihood of any area being there. But most of you would agree that this just does not really look like a brain map, right? You're expecting somewhat more of an atlas where you have borders between areas. So what we need is some form of a summary map that integrates all of this information. And we compute that quite straightforwardly by what is called the maximum probability map. The maximum probability map is a summary map of all examined areas, and each voxel is assigned to the most likely anatomical area at that position. Now, this is a somewhat older summary map of the U-Brain Atlas, but I think it represents some of the features quite nicely. So the key feature of this maximum probability map is that it produces a continuous, non-overlapping parcellation of the brain. So in that respect, it looks a lot like the old classical brain maps published by Brodmann, von Economo, and others. But there's one key distinction. So when you look, for example, here at the area between the border between area 44 and 45, this is not a typical or schematic representation of that border, but rather based on a sample of 10 different postmortem brains, it is, so to speak, the watershed, and on the right it was more likely to find area 44, on the left it was more likely to find area 45. But the stress I put on there already indicates that it's not an absolute border, so left to that border it's always 45, right it's always 44, but you have to acknowledge that we are in a probabilistic setting. We're talking about comparing usually group activation data in vivo with some group anatomical representation post-mortem, and unfortunately it makes the description a little bit less black and white than the Taylor-Ruck Atlas, which tells you it's area X, but rather now we have to acknowledge that it's most likely this area, but it can also be with a certain possibility another area. And I will show you some more examples of that. So, coming to the key question then in vivo imaging, where is my blob? And I will illustrate that in the next couple of slides. So the question really is we have this probabilistic maps, we have the maximum probability map as a summary, and we have some neuroimaging findings. And we need to bring these together, and if you want to use that as a standard tool, it has to be easy to bring them together. So we have created a software that's called the OHBM Anatomy toolbox that allows you to do that, and there's also implementations, very similar implementations of that in FSL as part of the FSL Maps tool and in AFNI. And with that, we can now investigate structure-function relationships. So what you see in red is a blob from a now very old neuroimaging experiment. It was just tactile versus visual simulation. and we got an activation on the post-central gyrus, which obviously one would expect. And the first thing you can do is the visual superimposition. So basically, in the background here in different shades of gray values, you see the different probabilistic, the different side diatonic maps like Brodmann area for A and B, the anterior and posterior part of the primary motor cortex, area 3A, 3B, 1 and 2, so the different areas of the somatosensory cortex. And you can already visually see quite well that this blob corresponds to the primary and then higher somatosensory areas on the postcentral gyrus. But then we can also put some quantification on the degree of overlap, and we can do that either based on volume or on local maxima labeling. What is volume labeling about? It's actually a very straightforward concept. So we consider the entire activated cluster as being a homogeneous thing. So we're not considering the internal differentiation like more or less activated, but it's above the threshold, so this is a significant activation. And then we compute the intersection of that activated volume with the maximum probability map representation of the different histological areas. And then we can express that overlap basically relative to the cluster volume. So here, about 2/3 of that cluster was located in area 3B. Or we can do it relative to the area volume. So in that case, only 2% of area 3B are activated. Now in that case, the very low number we get for the second is not surprising, because it's a somatotopically organized area, so the hand representation, in particular that task, should only recruit a certain smaller part of it. Now one question that comes up, I would say somewhat between frequently and very often, on the SBAM helpline, or people asking me directly, is like, what's the better measure? Which of the measures should I use? And there is no direct answer to that, because it really depends massively on the size relationship between your blob and your histological areas. So in that case, making the point on the percentage of the histological areas being activated is fairly useless. But you could have the other scenario where you have a rather large blob that, that spreads maybe across many different areas, but it recruits like 90–100% of these 2 or 3 areas. So really, which of the two information is more reasonable and more valuable really depends primarily on the size relationships. Now we can also do a local maxima labeling. Now the key difference between the cluster labeling, taking the entire activated volume, and local maxima labeling is that we now look at the peak voxels within that cluster. And for the peak voxels, we can say, we can ask, is that voxel assigned to a particular area? And if so, what's the probability? And what's the probability for other areas? And you quite nicely see here the somewhat more gray world of probabilistic mapping relative to the black and white you get from the Taylor rock demon, that yes, it's somewhat more likely that it's in area 2, but actually it's the same base probability. Now you could ask, why is it then assigned to area 2 rather than area 3B when both have a probability of 50%? Well, that's basically taking into account the neighboring voxels and breaking the tie by using the surrounding. And if it's not assigned, then we can ask which other areas are found at that position. Now, cluster or local maxima labeling, a second very popular question, which one is more useful, more telling? Well, they're both basically founded on different ideas on the functional data. So the cluster labeling says, this entire cluster passed the significance threshold, that's what I consider my activation. And it does have the very nice property that it is much more stable against smaller shifts, for example, due to spatial misnormalization. Because you're comparing a volume to another volume. If you shift by one voxel, you're not going to get too much perturbation. Now, the local maxima labeling is in particular useful if you have a larger activation with distinct sub-peaks in it. Then assigning the whole thing may not be all that useful. We want to say this peak is in that area and that peak is in another area. But since you're now basically assigning one particular voxel, the peak voxel, to a particular area. That also means that whenever things don't go all that well with spatial normalization, for example, shifting one or two voxels makes quite a bit more of a difference. So, how we can use that then to bring different things together in a probabilistic framework? We do have these probabilistic cytoarchitectonic maps, and really, by the way I've just showed you, you can integrate already a lot of different modalities in that framework. So anything which gives you voxel-wise results in MNI space is comparable. Whether it's lesion data with voxel-wise lesion symptom mapping, morphometry, fMRI, or even if you get data from electrophysiology that's in a voxel-wise MNI space. Anything of that can now be referenced against the same anatomical framework. And that's an example here. So we have these two activations here on the inferior frontal gyrus. It's about spatial in red and green temporal uncertainty coding. And now the big question is that actually one thing, or is that a different one? Because they're kind of close to each other. So one could argue about, well, this is just a little bit of variance, or it's actually two different parts of a brain. Now, if we compare that to the histological atlas, we actually see quite nicely that only the green, the temporal uncertainty coding, that is in area 44, whereas the red, the spatial uncertainty, is located just outside of area 44 on the ventral premotor cortex. So functional and anatomically distinct representations can be inferred from that. Another example from VBM data, so it's gray matter reduction in schizophrenia. And again, we can, using the very same approach, label that and attribute it to histological features. And I hope you start to see the biggest potential of that. Now here we have an attribution to a particular nuclei, like this primarily in the left lateral basal amygdala. Now, if you have other studies that use the same reference system, this anatomical allocation, then you can actually be sure that you're meaning the same thing. And you're not dependent on what you or somebody else thinks is the lateral basal amygdala, is area 44, and so on. Now, you can also go the other way. Now, a lot of what I talked about now is you have in vivo data, and you go to anatomy to tell you where it is. But you can also go the other way around and use the anatomy as a priori information and then infer some properties in vivo. And I think a very straightforward example is connectivity analysis using either diffusion tractography or functional connectivity mapping. So you can use anatomical priors as your seeds and then use that to assess task-free data. And here's an example of that we did a few years ago. So we used a seed mask here from an area in the secondary somatosensory cortex, and then used diffusion tractography to find out that particular anatomical region, where is it connecting to. And the nice thing again here, and I know I'm being a bit repetitive, but I want to stress that point is, we do know the equivalent of that area in non-human primates. And there's a ton of literature on non-human primates on invasive tracing. Now the nice thing is by using this histological framework, we know we are in the same area that people also investigated in primates, so we can make direct inferences about to what degree is the connectivity homologous between species or not. So finally then, we can also, for example, use receptor PET data, so use that as a region of interest. And as I already mentioned earlier, the nice thing is we have the link to anything which is ex vivo. So using the cytoarchitectonic definitions as our common language, we can now both investigate in vivo data, where we get the receptor density from receptor PET in a particular area, but we can also use that same area definition and look in postmortem data, where we can identify that area based on histological cytoarchitectonic sections and do the comparison between in vivo and ex vivo mapping of, for example, receptor densities. So I hope that this overview has given you an idea that it's about localization and that post-mortem cytoarchitectonic maps can be very useful tools for localization and describing. But above that, and I think way more important, it's also a way to integrate, because it basically provides a common language. If you talk about the histologically defined laterobasal amygdala, and you assign VBM data to it, you assign fMRI data to it, you use that as seeds for functional connectivity analysis, but you could also look into any post-mortem data like receptor architecture in that same area. You can link it to data from non-human primates that also did histological analysis and described their results in terms of anatomical nuclei. Then all of a sudden, it's more than just localization, but I think actually this common histological language is really a key towards a more integrative and more standardized description, and hence something I would call integrative neuroscience. And with that, I would like to close and thank you for your attention. Obviously, thanks everybody who's been to everybody who's been involved. Some of the people who did the anatomical mapping, and obviously Katrin and Karl, who pursued that idea for many years before I even started. Thanks a lot.
The talk's open for discussion. Ellen, yeah?
Simon, a nice talk as always. This whole notion of probabilistic cytoarchitectural fields turns on two things, two aspects of the nonlinear, of the mapping function that you use. The number of degrees of freedom, the dimensionality of the warping function that you use, and the features that you are using to drive the warp. They can be structural features, functional features, indeed they can be cytoarchitectural features. So could you tell us a little bit more about the decisions that went into those two aspects, which ultimately define the degree of probabilistic atlas you're getting?
Absolutely. And in fact, this is a question really not just, I think, to me and to us on the histology, but it's a question to the entire field. And I would argue that at the moment, we as a field, not just we as those people who do the probabilistic mapping in Jülich, are still at a stage where it's mainly high-dimensional warps based on gray values in structural imaging data. Now, it is a very, very good question if this is where we should end up, if that's the best we can do. But I could make the argument now at the present, not so much for the future, but for the present and the past, that this is what neuroimaging has used, and this also then should be what we use for the histology, just to be fundamentally comparable. Now, I absolutely take your implicit point that we should move on to refine our spatial warps. Surface-based methods, obviously, is probably one of the topics you had in mind. And I think there is a big field and constantly growing field of how you ideally normalize the data. And obviously, we, having to normalize our post-mortem data, have to deal with and interact with that field. We do have a little bit of the problem that the reconstructed post-mortem scans are not as, let's say, nicely normalizable than high-quality in vivo images. But I think this is a question that's sort of definitely more overarching than specifically looking at post-mortem in vivo comparison, as like what's actually the best way to normalize brain images? What do we mean by good normalization? And the pragmatic approach up to now would be to say we use basically what the in vivo field, the imaging field is using, and that's basically volumetric registration on gray values. But yes, looking at the surface-based methods and other features to drive registration will be an important part in the future?
Actually, this is a question which is probably for many of you interesting, and I might involve and ask David, for instance, what you would respond on this. So I think we can now really go over to a broader discussion and perhaps use this question, which Ellen has addressed, to provide also a different answer or an additional answer, not necessarily a different one. So thank you, Simon, at this point, and thank you for your talk. Yeah, and let's open now the last part, meaning that please comment, ask questions to the people who presented here, and perhaps also to comment some of the findings that have been presented here.
I guess to take Katrine up on her follow-up opportunity, I do think it is extremely important for the field to move forward in terms of endorsing, buying into the importance of surface-based registration whenever possible when studying cerebral cortex. As Matt has shown earlier today, and a number of groups have shown, the improvements in registration when using folding cues alone is substantial, and then what Matt touched upon and what we're now seeing from a number of studies, but including recent efforts from the Human Connectome Project, multimodal surface registration can really markedly improve intersubject alignment, particularly in regions that many of us care intensely about in terms of the higher cognitive regions where folding variability and area to folding alignment is more problematic. So these are becoming soluble problems, and I would definitely encourage the community to really work hard, including with postmortem data. That is a particular challenge, as Simon indicated, but those are not insoluble problems. So I would hope that will stay on people's radar screen.
Thank you, David. Perhaps we should mention today, we focused very much on the cerebral cortex. Yeah? So this is, our anatomy session was very much cortex-driven, so to say. And for going into the depths of the cortex and having, for instance, high-resolution data as Rainer provided, this spatial registration with respect to surfaces is a major advantage. When we think about the whole brain, of course, we have to find perhaps different strategies here. And as people are interested in the cerebral cortex, we cannot simply neglect, and of course we do not neglect, that there are some thalamic structures, amygdala, and so on and so forth. And when we want to understand the connectivity and the wiring scheme of the brain, we have to take it into account. And this, of course, again, requires for a little bit different strategies. So Ellen wanted?
Yeah, I certainly don't disagree with the utility of using surface-mediated mapping schemes, but my larger point I thought I was making was that if we're looking at registering functional neuroanatomy, we might be better off using functional landmarks rather than structural landmarks as the basis for the warping function. We were using functional landmarks many years ago to bring visual cortex into better spatial registration. If you wanted to look at subtle second-order functional areas around major lighthouses, Jim Haxby has now popularized the idea again with hyper-resolution and these ideas, but they're all boiling down to the same issue, using function to drive the warping rather than using structural features, if you're looking at functional neuroanatomy and cytoarchitecture.
Thank you. Rainer, you wanted to?
I would say I like this point very much. We recently have worked about a combined cortical, curvature, whatever you want, alignment together with functional markers to also have an answer to the debate, which was very popular from Carl Friston on the one side and Nancy Campish on the other side, a few years ago in Neuroimaging, and where Carl Friston argues if you do a whole brain analysis, functional one, you should not do any subsequent ROI analysis, whereas others say we can solve exactly what you said, the alignment problem, because if we first localize the regions and we align them, we basically have super powerful statistics because we have no spatial smoothing necessary and so on. And B said, why not combining a whole brain approach, cortical approach, with functional markers where they are available, of course, and that's the tricky part. I think when I talked to Carl and Katrin some about 10 years ago, I already told them and said it would be nice to do the same you did with your atlas, but at the cortical level. And I think what we see part of Kevin Weiner's work and also in our group shows when you use these small structures like the mid-fusiform sulcus, you suddenly get in the cytochronic maps, FG1 to FG4, almost 100% aligned, at least around these structures, and functionally the medial and posterior fusiform region also almost 100% aligned. I mean, much better at least as without that. That points to both aspects from Dave, that we should use cortical ones, but we should also add structures. This can be, as Kevin nicely said it, forgotten little anatomical structures, which might help to align functional and cytochronic areas better. And I think that would be a more complex alignment scheme, but in my opinion, way better than MNI alignment. For example, when I see the nice maps we have seen before, if you look alone in the maps around the motor and somatosensory cortex and see this 50% probability we saw at this place, if you would do a sulcal alignment, central sulcal alignment, this would solve this to a large extent because you have not the MNI half a centimeter variability where the central sulcus is. If you align it by cortical fold, you would of course increase dramatically the precision of these maps. But I think the ultimate goal, and this is really what I want to say, which integrates what we heard from Ellen and from Dave before, what I really think would be the coolest thing, which I would always dream about, is that we once understand how your cytoactinic areas link to the functional area so that we can really make precise maps, if this is possible, where it is possible. For example, what is FG1? It's too big to be an FFA area. It's way too big. That's my problem always with these things. But imagine at some point we would know that FG1 is FFA posterior, medial, and something else, I don't know. That we then can, of course, also at some point get with high field imaging, not cytonics, but at least some areal boundaries, like some people have already shown based on diffusion and the cortical diffusion, where they can see boundaries which seem to be similar to what you are doing. And if that would be possible in the human, then you could indeed use not functional data, but you could use the cytonic mapped areas in the human brain, like the myelin mapping we saw for those regions where this is possible, and then basically align the brain driven by cytotectonics. When you then know the mapping to the functional areas, you don't have to do in each subject all the experiments, but use some structural scans like a diffusion-weighted scan, if this is, I'm dreaming now, right? And then having areal boundaries and use them for alignment, and then do your specific functional experiments, and you could label them probably with a high precision. This is kind of a future I would envision.
I think we're getting closer than you think, actually, Rainer. So is this on? It is? Okay. So we've actually figured out a way to actually take the cytoarchitectonic maps from Carl and Katrine and actually using cortex-based alignment, project those back into individual subjects and just using cortex-based, in vivo subjects, and just using cortex-based alignment, the cytoarchitectonic maps match really perfectly to the cortical folding. So the MFS, you know, all the four FG regions flank the MFS, and we can actually see in vivo where the different face areas are, and the two fusiform face regions actually are in different cytoarchitectonic territories. And exactly what you're saying now, we can then do myelination mapping in vivo, see how that matches, if it doesn't match, do diffusion as well. And so we can actually, I think the point I'm trying to add is not only doing the atlasing work, but also then projecting that back into individual subjects to make sure that it matches the cortical folding of that particular brain. And then you can actually see and assess where any artifacts are in the process of the actual warping that we were touching on before. But we're getting, I mean, it's striking at how well these probabilistic maps of the cytoarchitectonic fusiform regions actually match really well to even individual subjects in vivo, and just to get that clarity. And of course, as you're saying, being able to do all the different functional mapping in vivo, and then also making those maps available to everyone else as well. And so then we can actually, you know, get to your dream of doing, or we have predictions from the postmortem brains of what the cytoarchitectonics look like, to then try and find ways to match that with in vivo methods and with MR. So I think we're getting closer.
I would like to make a remark, and coming back to Alan's question. Our final aim is not to match maps of different modalities. This is simply a technical question. I will not underestimate the technical problems. Our final aim is to understand how structure and function comes together. And the problem is that we cannot understand this just by integrating maps. Before we understand what is behind a function, what is behind the structure, and what drives the function and what drives the structure. So we are back to the very old question, how does structure and function interact? We always say it's two sides of the same coin, and then we escape the unpleasant situation to explain more this fundamental problem. I will make it more concrete. Let's imagine that we don't understand what somatotopy is, because it was mentioned during this educational course. We have an activation in the somatosensory cortex, somewhere in area 3B. Because we have stimulated the index finger of the right hand. And then we look into the cytoarchitectonic map and we find area 3B. And then we match both findings. And what we find out is a real mismatch, because our activation is so small. It is somewhere within 3B, but it does not cover 3B. Very simple. Now we know that it can be explained because of the somatotopy. We didn't have analyzed all the other nine fingers of the right and of the left hand and the arm and the rest of the body and so on, because we understood what is behind this activation. In many cases, we do not understand what is behind an activation blob in the cortex. That's what Rainer addressed, and especially in the higher associative areas. So I think we are far from having a good understanding of what a functionally driven brain map will really present us. We must really compare it. Yes, I agree. But this is not the solution, to use a functionally driven map as the ground truth, and then to adapt and to compare it with other maps. We have to do it, but the final aim is to understand what is the relation between the function and the structure. And we are far from understanding this, is particularly outside the primary sensory and primary motor areas. And even there, we have still to do a lot.
If I can amplify a bit on aspects of Carl's comments, which I resonate strongly with, I think there's a general point. One is, I and many others from comments in this today's discussions have implicitly assumed that the difference between one individual and another, particularly for the cortical sheet, is the size and configuration of cortical areas in a standard parcellation. An alternative, for which there's now growing evidence, is that the individual maps may have a particular area that is not only larger or smaller in one person versus the next, but the actual intrinsic topology may differ. And to the extent that that is a significant component, and I believe it is, then simple, or even the best surface-based alignment or the best volume-based alignment cannot compensate for that. So the kind of take that as far as it can and then use aerial classification methods of the type Carl Hacker and others have been developing methods, and Matt Glasser is now applying it to HCP data. That's going to increase the sensitivity of our ability to identify distinct areas in robust ways in individual subjects. A corollary of this, I would say in broad strokes, is the importance of getting more and better data from individual subjects rather than just getting bits and pieces of data from many subjects. A general message from the HCP approach is that 4 hours of imaging data across multiple modalities yields enormously rich rewards that could not have been replicated by parsing it into more subjects and smaller amounts of data. And as people design experiments going forward, then thinking about what's the best way to characterize and get a rich fingerprint of functional and structural organization and processing in individual subjects, I think there will be great value in swinging the pendulum towards more in each subject is better in general.
Thank you, David. I think this discussion is now coming to the core of the mission of OHBM. I mean, we must really think about what all this mapping, which we have developed over the last decades, over the last 2 decades, to such an attractive way of analyzing the human brain and the brain of other species. What does this mapping contribute to the deepest understanding of the brain organization? And then we can feel much more freedom to think about new strategies, how to analyze the data. And I mean, this includes also our cytoarchitectonic, receptor architectonic data, our connectivity data, and the functional data. And we need a solid basis, a solid technique. We must discuss advantages and disadvantages of surface-based mapping, volume-based mapping, and other things, and we must develop it. But we must always come back to the question, what was our initial interest in this study? What is the question we would like to answer? And I hope this educational course has stimulated these thoughts by comparing so different multimodal approaches to brain maps, which are only meaningful if you bring the multimodalities together to answer the question which drove you into this field of research.
Yeah, well, so this was also one of the motivations that we had to go into more detail with respect to the visual cortex, because this is perhaps one of the only or one of a few regions in the brain where we have this convergence of information. On the other hand, it's a little bit dangerous because the visual system is a very specialized system, and we leave a little bit away certain higher associative function, cognitive function, which are much more fuzzy defined and ill-defined at this present place. However, we were hoping that that you enjoyed anatomy and see that this is not just structure and localization, and hopefully this came out. Not only this from the talks, we would like to ask you and encourage you to complete the online evaluation survey to help us also to reshape the courses if necessary, or to encourage us to do it in a very similar way in the next years. And if you have no other question at this moment, I would like simply be happy to close this session and thank you for being here, for participating, for taking notes, and would be great to see you during the next days and answer questions, to come into discussion with you. Okay? Yeah. Thank you.